Hello there. In our recent bulletin highlighting the latest financial results from Volition, we mentioned the exciting news that they'd been selected to submit 3 abstracts to ASCO. That is the world's largest oncology conference. At that point, they were embargoed, but the embargo has now been lifted. So to talk to us in more detail about these abstracts, I am joined by Cameron Reynolds, who is the CEO of Volition. Cameron.
Yes, Nick, it's really good to be able to actually tell you what was in them all now. And all 3 of them were exciting in different ways and showed different things. So each one deserves its separate attention. But it's really good that the American Society of Clinical Oncologists, which have the biggest convention in oncology in the world every year, saw fit to approve 3 of our abstracts for publication. And also, we were one of the 10 companies to be featured in their virtual media room.
Okay, Cameron, so let's drill down into a bit more detail about each of those abstracts, starting with the one that deals with your Nu-Q capture program.
Our Nu-Q capture abstract was very exciting, as this is the very first data we've now made public on our capture program in its entirety. And it went through specifically how we can now concentrate long from short nucleosomes. We have a lot of things being developed under the capture program. A lot of different assays now are being developed. Just for background, that's where we pull out the nucleosomes rather than just labeling them in the kits. And that allows us to do a lot more investigation into those nucleosomes. So it's been shown for a few years now that long from short nucleosomes is interesting, because the short nucleosomes are much more likely to be from the cancer. So by pulling out the long ones, we have shown in actual cancer patients now, in colorectal cancer patients, we can concentrate the nucleosomes of the short, therefore the ones from the cancer. So very, very exciting. And don't forget the capture program, this is the very first application of it. This is the separating long from short nucleosomes, which we'll use in our assays and potentially licensing to sequencing companies, which should help with the sequencing programs and make them a simpler way to sequence. If you can decrease the noise and increase the signal, that should be very helpful. And then in a wide range of other uses. And all the other uses for capture, this is one structure out of the thousands on the nucleosomes we can concentrate for. We're working on a lot of others. So we'll have a lot more information on this aspect and on the other aspects in the coming months.
And that really deals with some cutting edge science. So you can find out more about the abstract in full by looking at it on the Volition website, or you can look at the video that accompanies that particular publication. Let's move next on to lung cancer, Cameron. And really here we're talking about one of the world's most deadly cancers.
Yes, Nick. Unfortunately, lung is a very deadly cancer, the deadliest. And part of the reason it's very deadly is, unfortunately, it's often diagnosed very late. That's partly a function that the current diagnostics are quite invasive, they're not very accurate in the whole, so it's a real tragedy. So there's a massive need for any type of blood test which can really help with this. So you'd have to say this data is very encouraging. In 142 samples from Liège Hospital, which is in Belgium, we're working closely with clinical partners here who are also as happy with how this is going as we are. We had 142 subjects, importantly including a competing condition, something that has symptoms similar to cancer but is not cancer, COPD. And really interestingly, we had a very good discrimination between the healthies and the cancer and also the competing condition of the cancer, 88 and 85% accuracy, which for a single or small panels is really quite special. So this is the first step. We're doing a lot more work, which I'll outline later, but a really good step in the lung cancer path to the product. And I think it's extremely encouraging, and our collaborators will now work with us to work on the next steps.
You mentioned the work going on in Belgium connected with that particular lung cancer abstract, but I understand you've also got a larger study that's been going on in Taiwan.
Yes, Nick, the studies in Taiwan are progressing very well. They're collecting about 1,500 patient samples, mostly lung cancer, and then some other healthies and competing conditions. So that's a really good trial where we'll have a lot of data from a great collaborator. Lung cancer is a real problem in Asia, even more than the rest of the world. It's great to have the collaborators working in Taiwan. And it's really good. The capture program is extremely good from a whole lot of points of view, but we're also making very strong progress in our actual assays, our marquee trials in lung and colorectal. And the data we've shown, plus the ongoing trials, as we've talked about, really show the progress we're making in our marquee big trials.
So let's move on to the third abstract for ASCO, and this deals with blood cancer.
As to blood cancer, it is yet another cancer where we've shown really good discrimination on a very small number of assays in these first proof of concept studies. This one was 116 samples, so a good mix of cancer and healthies, and extremely good discrimination in these liquid cancers. It was about 91% accuracy, which is quite remarkable. And for those who are not familiar with them, the blood cancers are also very, very deadly. And they lead to about 415,000 deaths a year. So very tragic. And because the symptoms are non-specific, you feel tired and other very common symptoms, it's very difficult to diagnose. So if we can show good clinical efficacy with this data, it's something which could be used very, very widely worldwide. So we're in the process now of adding blood cancer to our list of cancers which are the big marquee trials, in adding it to lung and colorectal as our big ones. And very interestingly, and this is something which we talk about, this is the first link to the vet side. This is one of the cancers which has shown up extremely solidly in the same assays, with the same assays, in the vet space, in dogs. I really could not be prouder of our scientific team. The data that we've now come to ASCO, which is a fantastic conference, we're now very aggressively getting out there and showing what we have. We think we can be really at the heart of epigenetics, and epigenetics is becoming more and more important as it becomes more and more understood how important it is. So to have great data in several cancers, as well as the very exciting capture program, couldn't make me prouder or happier with the course we're on, that we're doing the right thing, that we've got the right technology, and that we can add a lot of value to a lot of different areas in epigenetics and in healthcare going forward.
Thank you very much, Cameron. And as we mentioned there, all three abstracts are published on the Volition website, all with accompanying videos. And we look forward to bringing you more exciting news and data from Volition in the future.